Hello and welcome back to Biology for Beginners. This week we continue on with our march through chapter 13, How Populations Evolve, by talking about the evolution of populations. Imagine that. So without further ado, let's get into this. As always, right now the terms and definitions should be popping up somewhere around me so that you can either pause the video and grab the vocabulary we will be using today, or you can head on over to my Patreon. The link is down in the description below, and you can have the entirety of my slideshow as a PDF if you would so like, for absolutely free. However, if you do choose to make my day and become a patron, you will get all of my Bio for Beginner material at least a day in advance. So let's get into the topic at hand today. Before we dive into the new stuff, I know last week's video was kind of long-winded, so let's do a quick review. I promise it'll be quick. So, the theory of evolution states that modern species are descendants of ancestral species that changed over time. Natural selection is the driving force behind evolution, and it just is the idea that individuals with certain traits survive and reproduce more than individuals that do not have those traits. Adaptations are inherited structures that enhance an organism's ability to survive and reproduce in a particular environment. Evidence from multiple fields support the theory of evolution, and evolution has no end goal. Now that we've had that quick review to jog your memory about the word salad I gave you last week, let's get into the new stuff. So, one thing that evolution needs is genetic variation. And what exactly do we mean by that? Well, if you'll notice, physical traits among individuals are going to vary from one to another. The example we have here in this slide is a bunch of garter snakes. Now notice these 4 snakes all look very different, but they were collected in the same area, so they are representative of the animals living in that particular area and the variation between them. They vary from having that very bright red stripe, to being very spotted, to being kind of plain looking. But they are all garter snakes, and they represent variation among individuals. So the thing about the physical traits that you're seeing here is that they can be passed on from parent to offspring. This is because the genes that are going to be passed on code for those specific traits. And the reason we get variation like this is that there are often 2 or more alleles for a given gene. At this point, if you're feeling a little confused by the word allele or this whole concept of genes being passed on, please review the Mendelian genetics videos that we have put out. I believe it's chapter 9. That will really help you grasp the rest of this video. So if you're confused at this point, go back, do a review, and then come back to this. Moving on. So because we have these alleles, and the organisms that we're going to be talking about reproduce via sexual reproduction, we know that sexual reproduction is going to shuffle those alleles into new combinations, and thus giving us a lot more variation in the physical possibilities among these populations. Since sexual reproduction can only shuffle the alleles, we then have to ask the question of where do new traits come from? Where do new alleles come from? Well, they're going to arise through mutations, which are just changes in the genetic information of a cell. And this is the ultimate source of genetic diversity, because pretty much you can't get new traits if there's no mutation, because otherwise it's just reshuffling the same deck over and over again. And the only way to change it up is to bring in a new card. So now that we understand that for evolution to occur, we need genetic variation because we need to have alleles coding for these different traits, and then we need new alleles to pop up through mutation, we can begin discussing the question, where does evolution occur? Well, evolution is going to occur specifically within a population. And a population is just a group of individuals of the same species. However, there are two caveats to that. One is that these individuals have to be in the same geographic location, and they have to have that ability to interbreed, which kind of goes along with point one. So a population is a group of individuals of the same species in that same geographic location that are able to interbreed. So this group of bison represents a population of bison. They're all the same species, they're in the same location, and they have the ability to interbreed. And the next thing about the population that we need to understand is what a gene pool is. A gene pool contains all of the alleles in that population. So it's at every locus for every individual will be represented in the gene pool. Now, the little thing that I've come up with to kind of help you visualize that idea are, imagine these three color-coded circles are three individuals of our population. So our population size is 3. These organisms are diploid, so they're going to have 2 chromosomes, and each chromosome is going to have an allele for a gene. These particular organisms only have 3 genes, but notice that they have 2 alleles for each of their 3 genes. They have gene 1, which is represented by the pink or green triangles, gene 2, which is represented by the red or blue trapezoids, and gene 3, which is represented by the purple or yellow diamonds. Now, our organism 1, this kind of creamy circle on the left here, has 2 green triangles, a red and a blue trapezoid, and 2 purple diamonds. Those are the 6 alleles from this individual. So if each of those is one of the alleles for the gene pool, when we look at the gene pool, it's going to be all of the triangles, all of the trapezoids, and all of the diamonds. Those are all of the alleles available when we're thinking about the population breeding and expanding. So the gene pool is just the representation of all of the possible cards in your deck. One thing that we routinely see is that there will be changes in allele frequencies in a gene pool, and this is known as microevolution. So what we see here, taking our example from the same hypothetical population as the slide before, we had the 3 alleles for the green triangle, 3 alleles for the pink triangle, 1 allele for the red trapezoid, 5 alleles for the blue trapezoid, 4 alleles for purple diamond, and 2 alleles for yellow diamond. Now imagine there's some sort of pressure. Either something happened in the environment, or some organisms possibly moved out of the population. We will talk about this in a few minutes, but there's some sort of pressure happened. And so the frequencies of the alleles in this particular gene pool changed. And now we have still 3 and 3 for the triangles, but notice our red trapezoid allele disappeared entirely. And now all 6 trapezoid alleles that are available in the population are blue. And then for some reason, we now have 3 purple diamond alleles and 3 yellow diamond alleles. There has been a change in our allele frequency in this population, in this gene pool, and microevolution has occurred. So before we get more into the things that cause these frequencies to change, let's talk about Hardy-Weinberg equilibrium. So basically, we know that through sexual reproduction, that alleles are going to shuffle. And basically, allele shuffling alone is not going to substantially alter the genetic makeup of a population. So the allele frequency will remain the same. And remember here, when we're looking at the allele frequency, we're not looking at the phenotypes of the individuals. We're looking at their genetic makeups. So it doesn't really matter how many of the homozygous versus heterozygous individuals we have, because we're looking at the genetic makeup and how many of each allele are present. Let's look at this figure that was pulled from the book. I know that it's a little confusing at first glance, but we're going to walk through it right now because it kind of demonstrates this idea that with only the shuffling of alleles, the frequency is not going to change and you're not going to get that microevolution happening. So this figure is based on the idea of a hypothetical iguana population. Now, some individuals have webbed feet, some do not. And the genes that we see here are a very typical dominant and recessive situation. Again, if this is confusing to you, please go back and review the Mendelian genetics from chapter 9. It will make so much more sense if you go do that. So our genotypes here are capital W, which is homozygous for the dominant trait, capital W, lowercase w, which is the heterozygous individual that will also have no webbing, as no webbing is the dominant trait here, or lowercase w, and this is the homozygous recessive individuals that will have webbing. Again, you can completely ignore webbing versus no webbing right now, since the allele frequency doesn't care what the phenotype is. Allele frequency is just looking at the genes here and the alleles. So in this hypothetical population, we have 500 animals. 320 of them are heterozygous dominant. 160 of them are, 320 of them are homozygous dominant. 160 are heterozygous, and 20 of them are the homozygous individuals. Now, if we move down here, we'll get the genotype frequencies. So 320 out of 500 would be 0.64. So 64% of them are the homozygous dominant. And then 0.32 heterozygous, 0.04 homozygous recessive. And what we're going to do here, now that we have these numbers, is we're going to look at the number of genes in the gene pool. So since these are diploid organisms, you can take the total number of animals, which is 500, and multiply 2, because each animal is going to have 2 copies of the gene. So there are a total 1,000 genes for the webbing or lack thereof in this gene pool. So we take this number and we then apply it to these alleles that we see popping up here. So you'll take the 320 homozygous dominant, multiply it by 2, and we get 640, because these animals have 640 capital W alleles. And so we have 640 W. Then we take the heterozygous number, 160, and we don't multiply that by 2. What we do is it's 160 capital W plus 160 lowercase w, since these organisms have one of each allele. So you don't multiply that by 2, because it's just 160 of each allele since they're heterozygous. And then for the homozygous recessive, you do exactly the same thing we did for the homozygous dominant. You have 40 lowercase w here. And this will end up getting you the allele frequencies, because then we have this depicted here. You add that all of the capital W's together. So 640 plus 160, which ends up with 800. Divide that by 1,000. That is 0.8. And then 160 lowercase w's plus 40 lowercase w's is 200. Divide that by 1,000. You get 0.2 lowercase w's. And those represent our allele frequencies in this population. 80% of the alleles in this population are the dominant allele, and 20% are the recessive allele. Now, we can move on to predict what the genetic makeup of the second generation is going to be. Now, since gametes are going to reflect the allele frequency of the parental gene pool, we make a happy little Punnett square. And we see here that the eggs are going to be about 80% dominant and 20% recessive, and same with the sperm. And then we just move across like you would for any normal Punnett square, figure out what we're doing here, whether it's homozygous or heterozygous. And we talked about this back when we were doing Mendelian genetics, so this should all be review here. But instead of just saying, oh, I have 1 to 2 to 1, we take our allele frequencies and multiply them together here. So at this homozygous dominant as an example, we take 0.8, multiply it by 0.8, and end up with 0.64. We do the same thing for the two heterozygous individuals, where we multiply 0.8 to 0.2, end up with 0.16, and then the same for the homozygous recessive, where our result is 0.04. So the genotype frequencies that we end up with are 0.64 homozygous dominant, 0.32 heterozygous, because we have to add 0.16 with the other 0.16, and then 0.04 of the homozygous recessive. So those are our genotype frequencies. And then we do basically the same thing that we did before to end up with our allele frequencies, which are going to be 80% dominant and 20% recessive. Notice allele frequency did not change despite them shuffling around through sexual reproduction. But also notice here that we can take what we learned from the Punnett square and figuring this out organically to plug into what is known as the Hardy-Weinberg Equation, which is p squared, which is the frequency of the homozygous dominant allele, plus 2pq, where pq represents the frequency of the heterozygotes, plus q squared, which is the frequency of the homozygous recessives. And that is going to equal 1 if we are at equilibrium. This equation can be used to determine if a population is experiencing microevolution or not, because if it is not experiencing any form of microevolution, it may not be exactly 1 when you do the math, since life is messy, but it's going to be damn close to 1. So that's the main use for this equation. But what are the 5 conditions that a population needs to be under in order for it to be at this equilibrium? And keep in mind that these conditions are very rarely met. The first condition is that the population needs to be very large. And the reason for that is because smaller populations are more likely to show kind of amplified effects of migration or natural disasters, things like that, all of which we will go over in the next video when we talk about some of the reasons behind evolution, all that fun stuff. The second kind of condition for this is that there has to be no gene flow between populations. So you can't have population A and population B mixing at all, because that would change the allele frequencies. And we'll go into what gene flow is in the next video. Condition 3, there can be no mutations. Remember, mutations give rise to new alleles. So if you throw a mutation into the mix, guess what? You just changed your freaking alleles frequency. You're not at equilibrium anymore. 4, it has to be entirely random mating. So every single male has to have an equal chance of mating with every single female and vice versa. Again, in the next video, we'll go over a little bit more why that doesn't really happen that often in everyday life out there in the wild. And finally, 5, natural selection has to not be working on the population. And that's probably the hardest condition to meet, because for the most part, natural selection is pretty much always going to be working on a population in some way or another. But even though populations are very unlikely to ever truly be at equilibrium, the equation itself is still useful. And the field of biology specifically has quite a few of these kind of hypothetical scenarios that serve as a template for us to be able to look at life and put it into a system and make models to predict things. But it's kind of like the ideal that's never going to be met, really. But that's all I have for you today. I know that this video has been kind of technical, especially going over the Hardy-Weinberg stuff, but it's not that scary. And most entry-level biology classes are just going to want you to recognize how to plug in the P's and Q's into the equation, and they want you to understand if the population is or isn't evolving. So if you plug everything in and it equals 1, you're at equilibrium. If it doesn't equal 1, you're not at equilibrium. Evolution's happening. So that's probably the biggest thing to keep in mind from that portion here, and know the 5 conditions to be at equilibrium. That's a big deal too. But I will now release you. Have a wonderful rest of your day, afternoon, evening, what have you. Don't worry, I'm not going to assign you any homework today. I promise. I'm the cool teacher. And I will see you all in the next one.